Welcome to the fascinating world of biomolecules. Imagine trying to write a massive, highly detailed encyclopedia containing all the instructions for building a human body. You would need an alphabet. In the biological world, nature uses a simple chemical alphabet of just 5 primary letters to write the genetic code of every living organism. Today, we will explore these letters, known as purines and pyrimidines, and understand why their unique shapes make life possible. To begin our journey, we must first understand what these molecules actually are. In biochemistry, purines and pyrimidines are classified as nitrogenous bases. This simply means they are alkaline molecules that feature nitrogen atoms embedded directly within their carbon rings. These specific ring structures are incredibly important as they form the very foundation of how our cells store and transmit information. Without this unique chemical arrangement, the complex biological systems that sustain life would not be able to function properly. Now that we know what nitrogenous bases are, let us look at the simpler of the two families, the pyrimidines. A pyrimidine consists of a single, compact, six-membered ring made up of 4 carbon atoms and 2 nitrogen atoms. The primary pyrimidines found in living things are cytosine, thymine, and uracil. A helpful trick for students to remember these specific bases is the mnemonic cut the pie, which stands for cytosine, uracil, and thymine. This simple memory tool will help you easily identify them. Moving on from the single ring structures, we encounter the purines. Purines are larger and more complex than pyrimidines. They consist of a distinct double ring structure created by fusing a 6-membered pyrimidine ring to a 5-membered imidazole ring. The primary purines are adenine and guanine. You can easily remember this using the mnemonic pure as gold, where pure stands for purines and as gold reminds us of adenine and guanine. Before we move on, let us see this with a concrete visual example. On the screen, you can clearly see the structural difference we just discussed. On the left is the pyrimidine, a single, compact, 6-membered ring. On the right is the purine, a bulkier, double-ring structure. Notice how the nitrogen atoms are embedded directly into the carbon framework of both molecules. This specific geometry is what allows them to stack and pair perfectly within the DNA double helix. Having seen their structures, you might wonder why these specific rings matter so much in real life. Their most famous role is acting as the rungs of the DNA ladder, storing our genetic information securely for decades. However, their importance extends far beyond just DNA and RNA. For instance, adenine is a core component of ATP, or adenosine triphosphate. ATP is the universal energy currency that powers every single cell in your body. Without these bases, life simply could not function. Knowing how crucial these molecules are, it makes sense that they must be incredibly stable. Both purines and pyrimidines achieve this stability because they are aromatic compounds. This means their atoms share a cloud of electrons that freely circulate around the flat ring. This shared electron cloud acts like a strong chemical glue, preventing the molecule from easily breaking apart under biological conditions. This stability is exactly why our genetic code does not spontaneously degrade. This concept of stability leads us to a mathematical principle known as Huckel's rule. We can mathematically prove if a ring is aromatic using the formula E equals 4n plus 2. Conceptually, this formula determines if the electron cloud is perfectly balanced. Here, E represents the total number of pi electrons, which are the specific electrons participating in the double bonds of the ring. The symbol N represents a non-negative integer, like 0, 1, or 2. For a molecule to be exceptionally stable, its pi electrons must fit this equation perfectly. If the number of electrons increases or decreases such that N is no longer a whole number, the molecule loses its stability and becomes highly reactive. Now that we have our formula, let us apply it to a concrete example. Let us prove mathematically why the pyrimidine ring is so stable. First, we need to find E, the total number of pi electrons. A standard pyrimidine ring contains 3 alternating double bonds within its structure. Since each double bond contributes exactly 2 pi electrons to the ring, we simply multiply 3 by 2. This gives us a total of 6 pi electrons. Counting these correctly is the absolute foundation of applying Huckel's rule. So we have established that E equals 6. This brings us to the final step of our proof. We substitute our calculated value of 6 into Huckel's formula, setting up the equation 6 equals 4n plus 2. We subtract 2 from both sides to get 4 equals 4n, and then divide by 4 to isolate n. This gives us n equals 1. Because 1 is a perfect integer, the pyrimidine ring perfectly satisfies Huckel's rule. This mathematical proof confirms exactly why nature chose these highly stable molecules to store our genetic code. Now that we understand their individual stability, let us look at how these bases interact. In a double-stranded DNA molecule, these bases pair up across the helix. Because of their physical sizes, a bulky double-ring purine must always pair with a smaller single-ring pyrimidine. This ensures the DNA ladder maintains a uniform width throughout. Specifically, adenine always pairs with thymine, and guanine always pairs with cytosine. This strict pairing is crucial for the structure of DNA. This strict pairing behavior we just discussed leads directly to a mathematical certainty known as Chargaff's rules. The formula states that the percentage of adenine perfectly equals the percentage of thymine, and the percentage of guanine perfectly equals the percentage of cytosine. If the amount of one base increases, its corresponding partner must also increase to maintain the structural integrity of the DNA double helix. This simple but powerful rule allows us to predict the exact composition of any DNA sample. Let us see how this formula works in a real-world scenario. Suppose a forensic scientist analyzes human DNA and finds that exactly 22% of the bases are adenine. We need to find the percentages of the other 3 bases. We start with what we know. Adenine always pairs exclusively with thymine. Because of this one-to-one relationship formed by hydrogen bonds, an equal amount of thymine must be present. Therefore, we can immediately conclude that the percentage of thymine is also 22%. Now that we have found thymine, let us calculate the remaining bases. Since all 4 bases must add up to a total of 100%, we first add our adenine and thymine percentages together, giving us 44%. We subtract this from 100% to find that 56% of the DNA remains for guanine and cytosine combined. Because guanine and cytosine must also exist in equal amounts, we simply divide that 56% perfectly in half. This reveals that the sample contains 28% guanine and 28% cytosine. We have covered a lot of ground today, so let us review the most important ideas. First, purines and pyrimidines are the essential nitrogen-containing rings that form the alphabet of life. with purines being double-ringed and pyrimidines single-ringed. Second, their aromatic nature provides incredible stability, protecting our genetic code from degradation. Finally, their strict mathematical pairing rules ensure that life can be reliably passed down from one generation to the next without losing the instructions that make us who we are. Great job following along.